So let's get started. Hi, I'm Casper Mossman. I'm a communications director at QB3, and with us today we have Michelle Arkin, who is Thomas William and Frederick John McWilliam Distinguished Professor at UC San Francisco, a named professorship that comes with being chair of the Department of Pharmaceutical Chemistry. She also co-directs the UCSF Small Molecule Discovery Center and earned her PhD in chemistry from Caltech. Whenever you know that somebody went to Caltech, like they're scary smart. So anyway, I've known a few. Dr. Arkin's lab develops innovative approaches to screen for chemical tools and drug leads using biophysical approaches like fragment-based drug discovery and biological approaches, including high content imaging. Her lab's goal is to demonstrate druggability of new target classes and use compounds to discover new targets for drug discovery, areas of interest, including protein-protein interactions. We're going to hear a lot about that today. And allosteric and scaffolding sites in enzymes, focusing on diseases of aging like cancer and neurodegeneration. Dr. Arkin's also co-founder of Elgia Therapeutics and Ambigon Therapeutics. Really great to have you here today.
Oh, thank you. Thanks very much, Casper, for the introduction and the invitation. So shall I share my screen?
Please do.
Okay, great. So looks good. Everybody can see?
Yes,
Casper?
definitely.
Yeah, great.
Yeah.
Thank you. Okay. So thanks again for the invitation. And the title at the top today is, I don't know, it's kind of a little vague, meant to be provocative. How can we modulate the output of protein networks? So protein networks really drive biology, drive information transfer from one part of the cell to the other, or from outside the cell to inside the cell. So how can we modulate the output of these complex networks by targeting the protein-protein interactions that make them up? And as Casper mentioned, I'm a professor and chair of pharmaceutical chemistry. I'm also co-director of the Small Molecule Discovery Center. So the SMDC seeks to develop innovative and professional level collaborations in drug discovery and chemical biology approaches. So there we'll do high throughput screens, medicinal chemistry, other ways of finding molecules for biology that's of interest to collaborators. And we collaborate broadly around that. And then the research in my own lab has a few threads to it. One of them is this question that I've had for a long time, which stems from the observation that there are more drugs for some types of proteins than other types of proteins. And if you look at this graph, for example, if you consider all the drugs that we have that are approved for human use, about a quarter of them target this one family of transmembrane proteins, the G protein coupled receptors. And these are very common drugs, like a lot of heart medications, beta blockers, allergy medications, Sudafed, target this class of molecules. Very important in biology. But nevertheless, there are a lot of other classes that are important in biology. Then we have a number of drugs that target nuclear hormone receptors, and we get rarer and rarer and rarer until we get to protein-protein interactions, which are a tiny, tiny fraction, just a couple of drugs that target the interactions between two proteins. And the question is why? And it's not because protein-protein interactions aren't important for biology, because they're very important for driving biological signals. So thinking about it for many years, many people have thought about what makes protein-protein interactions so-called undruggable. When we talk about undruggability in this context, really, we mean unligandable, or difficult to find a molecule that sticks to the protein and has a functional effect. And thinking about this problem for many years, the field has come to these two basic ideas. That one thing that makes protein-protein interactions difficult to target is the flatness problems, that they're kind of flat. And here are two proteins shown in pink and purple. And you can see that where they come together, it's two helices coming together across a pretty flat surface. So where would a molecule stick on the purple or the pink protein to block that interaction when the two proteins themselves interact over a long surface area? And looking at that from the standpoint of a small molecule discovery, so a molecule is going to be smaller, is going to be like this size, in part of the interface, we can look at it like a cliff wall. And the cliff has little nooks and crannies in it. And that's how this woman is climbing this cliff by carefully positioning fingers and toes at just the right position in the rock. So if we can make molecules that find these little subsites of protein-protein interactions, and then we can scaffold those interactions very precisely, then maybe we can target even this flat interface with a drug-like molecule. The other side of the problem is essentially the opposite, the disorder problem. Many proteins fold upon binding to each other, and when they're not bound to their cognate proteins or receptors, they're intrinsically disordered. They're bound to scaffolding proteins or to chaperones that help keep them from falling out of solution and aggregating in the cell. But when you think of an intrinsically disordered protein from the standpoint of small molecules, again, it looks like this spaghetti and there's no place to bind. So we can just see that even these little, peppers just kind of sit on the surface. There's nowhere to bind to this intrinsically disordered spaghetti. So how can we address these two kinds of problems with small molecule drug discovery? So let's start with the flatness problem. This is something that we've thought about for longer. And an approach that has worked for the flatness problem is fragment-based drug discovery. So remember I mentioned that proteins at protein interfaces have these little binding subsites, like this rock climber can put her fingers and toes. So similarly, if you look at the surface of a protein, it has a number of little subsites spaced at some interval from one to the other. So if we try to take make a bunch of molecules that look like this, a bunch of linked pharmacophores or linked binding elements, without any knowledge of what we're trying to bind to, and we screen them, that would be high throughput screening, the chances that we'll find something that binds productively, but nothing that either binds unproductively or just wastes space, limiting the efficacy of this compound, it gets very difficult. So we're very unlikely to find a molecule just in the world that binds to several subsites. If instead, in a fragment-based approach, we break these molecules conceptually up into these individual binding elements, and we screen these binding elements one at a time. Now, this binding element doesn't need to know what else is around it. We just find and optimize this binding element for this site. Then we find and optimize a second element for a second site, and we link them in a later step. That's the concept of fragment-based drug discovery, and it works well for complex surfaces. So the approach that was developed when I was at Sunesis Pharmaceuticals for fragment-based drug discovery that's been really successful, that we've been practicing at UCSF as well, is a site-directed approach called disulfide tethering. And in this disulfide tethering approach, we have a protein with a putative binding site. I'm just showing one here, but you could imagine several sequential binding subsites. Next to this binding site, we have a cysteine residue. So we have a free thiol or thiolate. We then mix this protein with a library of molecules that have these disulfide bonds. And we do this under conditions that allow thiol disulfide exchange. We can allow this to come to equilibrium, and at equilibrium, we'll favor molecules that bind to the protein, that are complementary to the protein surface, and also have the geometry to make this disulfide bond. And we detect those covalently bound fragments by mass spectrometry. So here in this cartoon, we have the mass of a protein that's unmodified, and then the mass of the protein modified with this covalent fragment. A couple of the advantages of this technique for the projects I want to talk to you about today are that it's site-directed. So because we use the thiol, the cysteine residue, we put it near the binding site, or it naturally exists near the putative binding site, we can choose the location that we want our fragments to bind. So if you think about this, I said that there are several sequential binding subsites. It would be nice to be able to map those subsites one at a time prospectively. Second, we can saturate the binding site. So these molecules I mentioned, fragments, are these single binding elements. Because they're small molecules, they tend not to bind with very high affinity. There are not a lot of interactions that they can make. So having the covalent bond, the reversible covalent bond, allows us to increase the apparent affinity of these fragments. And because this is a tunable bond, this disulfide is reversible and it's dependent on the reduction potential of the medium. We can change the strength of that disulfide bond to get the correct level of sensitivity for finding fragments. And then we can saturate that binding site, which helps us screen for function. Instead of just for binding, we can also screen for change that this molecule causes in function. It also helps us with structural biology. The library itself has two components. It has this fragment component. So this is a diversity element. And then it has a set of linkers that vary in their length and their geometry and their geometric constraints. So because we're starting from a specific fishing pole and fishing into the lake, the distance, the shape and size of that fishing pole matters as much as the shape and size of the binding element itself. So here's an early example from Synesis where we use this fragment-based approach to find really some of the first potent inhibitors of a protein-protein interaction. And we had some molecules that bound to the surface of a protein called interleukin-2, and we were just having trouble getting higher affinity out of this molecule. So we reasoned maybe there's a binding site next to this molecule, and if we can use the site-directed approach to find these, a fragment that binds there, then we can link them. So this is the concept of fragment-based linking. And that's what we did. We identified fragments that tethered off of this engineered cysteine residue, and they were all these small aromatic acids. So from modeling, we reasoned that we could attach this aromatic acid to the green compound at this carbon atom. We made a small library. This was Brian Raimundo's lab, made a small library. And lo and behold, most of the aromatic acids were much more potent than the compound we started with. And the most potent is this furanoic acid, now coming in with a KD of 60 nanomolar. So this was a big breakthrough in affinity and drug likeness and in teaching us how to work with these kinds of protein interfaces for small molecule discovery. And there are many important things that have been discovered about the system over the years, but I just want to point out this one. I talked about the flatness problem, but when we look at interleukin-2 and we look at the surface of this protein and how it changes in crystal structures when different molecules are bound, it becomes clear that it's not really a cliff face so much as it's a little bit softer, maybe more like hay bale. So you can push on different parts of that molecule. And what you see in the crystal structure that looks flat actually has regions of adaptability that can curve around the molecules, where the molecules can trap low energy conformations in the protein. So when we look at, as we evolve this molecule towards higher and higher potency, we're trapping different conformational states of interleukin-2. And this turns out to be a pretty general phenomenon, that small molecules, that protein-protein interactions have regions of adaptability, structural adaptability, and that molecules can trap deeper, more small molecule-like binding sites, more deeper sites than you see in the protein interface itself. So we've used this technology over the years. Here's the interleukin-2 story to find non-covalent drug leads and then also covalent drug leads. So some of these molecules here quite potently and selectively inhibit otherwise undrugged targets, so brand new targets, by taking advantage of the cysteine residues that are positioned near functionally important sites in the protein. And the most famous of this is from Keyvan Shokat's lab, here looking at a covalent modifier of G12C in KRAS. So this is a potent oncogene. One of the oncogenic mutations turns residue 12 into a cysteine. He used our library to identify compounds that bound to that cysteine residue, really invigorated this drug discovery effort for RAS. So what are we going to do with it now? So there I showed you how we and others have used this technology to address the flatness problem. But what about the intrinsic disorder problem? So we reasoned that we could target the intrinsic disorder problem by stabilizing the intrinsically disordered protein bound to another protein. Because these disorder domains fold upon binding, in a sense, you take the spaghetti, when it binds to other proteins, it folds up, and here we have this little 3-helix bundle of fusilli. And now we have a highly structured, ordered complex, and perhaps we can find holes or pockets in this protein complex that we can bind our molecules. So small molecules can now trap these complexes through what we call a molecular glue mechanism. So we have a protein-protein interaction, here the gray and the green proteins, and we use a molecule that binds to both sides of that interface, to both the gray protein and the green protein, and holds them together. And in this way, we can modulate the function of that green protein that's otherwise intrinsically disordered. So there's another reason. So the intrinsic disorder problem is one reason to target stabilizing protein-protein interactions. The other reason is that proteins don't really act like these two isolated proteins like I've been describing, like this couple waltzing alone on the dance floor, but biology is really much more like these complex English country dances where proteins are interacting with many other proteins in a highly regulated and coordinated way. And if we want to understand the importance of each interaction in this complex dance, we want to think about how we can get specificity for a particular protein-protein interaction when these proteins interact with lots of other proteins. So looking at that in a more scientific way, we look at a protein-protein interaction network diagram here where each circle is a protein and the lines are called a node, and the lines between them are edges or interactions between two proteins. So here, for example, we have this protein that interacts with many other proteins shown by these multiple lines. And rather than knock out this protein, so one thing we can do to understand the function of this protein in this network is to knock out, to delete the protein. If we do that, we're going to remove all of its edges, all of its interactions, and we may collapse the whole network because this protein may behave differently in the absence of this interaction, and so may this, and so may this. So we can get a very broad view of the importance of that protein, but not a specific view. If instead we take this protein and we can just modulate the edge, just that one interaction, for example, by stabilizing that interaction, then we can understand the function of this protein-protein interaction in the context of this whole network. And so that's what we set out to do. Now, protein stabilization is kind of an unusual thing in drug discovery until recently, but it's a pretty typical way for natural products to behave. So here's a natural product, rapamycin, and rapamycin's function is to bind to this protein that induces binding to another protein, and that protein is then inhibited. So the function of rapamycin is to create a new protein-protein interaction that wouldn't otherwise occur without that molecule. We call that a synthetic or a neomorphic interaction. And the function of that neomorphic interaction is to inhibit the function of the protein. Fusacoccin, by contrast, is another natural product, a fungal natural product, and its job is to bind to this blue protein 1433 in the presence of this 1433 client protein, an ion channel, and ATPase. And when it binds to and stabilizes this protein-protein interaction, it has the function of activating the function of this ion channel. So fusacoccin stabilizes a native interaction, not a neomorphic interaction, and it has the function of activating the green protein rather than inhibiting it. So these two examples show us that by stabilizing protein-protein interactions, we have a lot of chemical biology we can play with, neomorphic or native, inhibitory or activating. There are some small molecules that also function by stabilizing protein-protein interactions, lenalidomide being the most famous. This is what we call a molecular glue degrader. It binds to a ubiquitin ligase, induces the binding of a transcription factor, and then that ubiquitin ligase ubiquitolates this transcription factor, leading to its degradation. So this is a very small molecule that creates a very complex set of functions, and it is a neomorphic interaction. So these proteins wouldn't interact otherwise. The integrated stress response inhibitor came out of a collaboration with Peter Walter's lab. This molecule, fascinatingly, functions and it inhibits the stress response in cells by stabilizing this decameric complex. It's a C2 symmetric molecule, and it binds in this C2 symmetric decameric complex and holds it together and makes it insensitive to upstream signals. So these two molecules are very fascinating, important molecules in biology, but they were both discovered from phenotypic screens, so from their effect on biology, and then only later was it understood how they function. So we would like to flip the tables on that and say, how can we prospectively design protein-protein interaction stabilizers? So we want to start with a system that we can tune and demonstrate that we can use a technology that systematically stabilizes just the protein-protein interactions that we want to. So we started with this protein 1433. Again, this W-shaped protein. It has this natural product, fusacoccin, that already does that kind of a function for a subset of targets. So we can take inspiration from this. So what is 1433? It is a very underappreciated protein in biology. It's a central effector of serine and threonine phosphorylation. So it has a network of maybe 1,000 interacting partners. So it binds to many proteins in mammalian biology that are phosphorylated at serine or threonine residues. And it's very often the reason or the effect of that phosphorylation comes from binding to 1433. 1433 binding can activate or inhibit the function of that protein depending on where it binds to the protein and the context that the protein is in. And many of the targets of 1433, many of its clients are these so-called undruggable targets, and many are intrinsically disordered. So one reason that we started working with 1433 is because our collaborator and good friend, Christian Ottman, is the world expert in the structural biology and chemical biology of 1433. He solved this structure as a graduate student and has been working on this target class ever since. So 1433, I think it's really a fascinating protein. You can think of it like a taxi cab, where you raise your hand and the taxi comes and picks you up and takes you where you want to go. So the effect of 1433 binding to the client protein resides within the client protein. So for example, 1433 can bind to a target protein, alter its conformation, which alters its enzymatic activity. It can bind to a target protein and occlude a nuclear localization site so that it translocates it to the cytoplasm and other things like that. So you can regulate subcellular localization, inhibits protein-protein interactions, and also it protects the phosphorylation site from being dephosphorylated by a phosphatase. Finally, it also functions in scaffolding protein-protein interactions, so it can also induce protein complexes. And some of these complexes that are induced by 1433 initiate the degradation pathway. So in a sense, they act like a degrader. They induce proximity of a protein with the degradation machinery. So our goal is to, given this range of biology and the range of important therapeutic targets that are bind to 1433, our goal is to systematically stabilize specific 1433 client complexes to block various disease processes. And so it's our example of taking this spaghetti, its folded state bound to its scaffolding protein, and using a molecular glue approach to fix this protein complex together. Now, the data that I'm going to talk to you about uses peptides as models for the client proteins for 1433. So this is a cryo-EM structure of a kinase BRAF bound to 1433 shown in white. Now, making this protein is a lot of work, getting specific phosphorylations, very challenging. So instead, we work with the peptide that's derived from the binding site of this protein for 1433. So let's zoom in on that. So in dark blue is the peptide that we'll use to mimic the protein-protein interaction. And you can see in light blue that it faithfully mimics that region of protein in the full protein. So the light blue is the full protein, just excising that piece of the structure, and the dark blue is the peptide itself, okay, both crystal structures. So we're going to, for biochemical assays and for structural biology, use the peptide as a mimic of the protein-protein interaction. And then when we go into cells, then we'll have the full-length proteins, and we'll be looking at effects on the full-length proteins in cells. Okay, so I mentioned that there are 1,000 targets. What makes me think that we can get specific stabilization of different protein-protein interactions when there are so many? It's these crystal structures that suggest that we can get specificity. So when you look at various peptides bound to 1433 shown in white, you can see that they scope out different sizes, different shapes, and they also have different amino acids pointing into the various binding grooves. So that natural product I talked about, fusacoccin, binds here. So there's a set of targets that, like estrogen receptor, where the C terminus of the whole protein binds to 1433, and the penultimate residue, here shown in yellow, is phosphorylated. So when estrogen receptor binds to 1433, it leaves half of the groove empty, and fusacoccin fills that space and stabilizes that interaction. Other proteins like P53 bind near the C terminus of P53, but the peptide takes a steep turn out of the groove, leaving a different size and shape to that groove with different amino acids pointing into it, so our molecular recognition features will be different. And then still other proteins like the transcription factor YAP binds through that groove. Okay, so the structural diversity is what suggests that we can get selectivity for small molecules. I just want to show you a little bit of an example using estrogen receptors, the example of how 1433 can work. So here, estrogen receptor, when it doesn't have estradiol, it's hormone bound, is bound to the chaperone HSP90. Then when this little white pentagon, estradiol, enters the cell and binds to the receptor changes conformation, dimerizes, enters the nucleus, and mediates transcription. However, when it's in this new conformation, this ligand-bound conformation, it can also be phosphorylated at the C terminus. If it's phosphorylated, it becomes a client for 1433, here shown in medium blue, and this blocks the binding to DNA. We can see that in this live cell image where estrogen receptor is in the nucleus normally. Then when we add a fusacoccin-like stabilizer that stabilizes this complex, we get more of the estrogen receptor in the cytoplasm, and you can see that quantified here. So the nucleus is shown in blue, and then green is some nuclear and some cytoplasmic localization of the receptor. This relocalization leads to inhibition of cell growth, inhibition of transcription, and inhibition of cell growth. This over time is looking at cell growth of MCF7 cells in the absence of any 1433 stabilizer. In the presence of a 1433 ER alpha stabilizer, we get a reduction in cell growth, and that's comparable to the ER degrader, estrogen receptor degrading compound called fulvestrant. Okay, so this is a natural product, and we're going to develop small molecules and show that our small molecules can behave like this natural product in inhibiting estrogen receptor activity. Okay, so how do we find those molecules? So we use this disulfide trapping approach now for the first time to stabilize protein-protein interactions instead of inhibit them. So here's 1433 again. Here's the estrogen receptor peptide shown in green, fusacoccin, the natural product, shown in yellow. And now the residues, here's a zoom. The residues that are colored are a wild type cysteine residue, cysteine 38, and then two other residues that we mutated to cysteine. And here's the very tip of the estrogen receptor peptide. Okay, so we're looking for molecules that bind in the fusacoccin binding site, and we're going to use the disulfide tethering experiment that I described, but we're going to do it in a new way. We're going to look for fragments that bind to 1433, but we're also going to look for fragments that bind to 1433 in the presence of estrogen receptor. And we want to find those fragments that bind better in the presence of estrogen receptor than in the absence. So if the peptide stabilizes the small molecule, stands to reason that small molecule binding will stabilize the peptide protein complex. And we measure those with 3 different types of experiments. First, we use the mass spectrometry approach. We titrate in that fragment and we measure its binding by mass spectrometry. And we can see for this particular fragment that it binds to estrogen receptor in the binds to 1433 in the absence of estrogen receptor a little bit, but it binds much better, these green dots, in the presence of estrogen receptor. Now we flip the script. We have estrogen receptor peptide labeled with a fluorophore. And now we're going to measure the binding of that peptide to the protein as we titrate in the compound. So here again, titrating in that compound and looking at the amount bound of the peptide. We can see we increase the compound, it makes the peptide bind, which we can see by an increase in fluorescence anisotropy. Then finally, we see what is the effect of the binding affinity, effect of the compound on the binding affinity of that peptide. And we see this here in these dose response curves. The peptide binds to 1433 with about a binding affinity of 2 micromolar in the absence of any compound. In the presence of our stabilizing compounds, we increase the binding affinity 40-fold. So now it's in the low nanomolar range. And how does it do that? Well, it's this tiny little fragment. It's really just a functionalized benzene ring, functionized phenyl ring. And it binds to 1433, shown in white, through this halogen in the back of the molecule. And it makes contacts, these edge-on contacts, with the estrogen receptor peptide itself. So it acts like a little bridge, stabilizing the peptide binding to 1433. So I showed 3 types of peptides before. I showed this truncated one that turns, and one that's straight. But when you look at several different crystal structures of proteins and peptides bound to 1433, you see that they really scope out a wide array of conformations. This is the fusococcin and our compounds binding site shown here. So we wanted to know what is the scope and limitations of our technology for finding stabilizers in the context of many different kinds of peptides. So we started here with these 5 peptides, the estrogen receptor that's truncated, a peptide that's turned, and 3 that are longer that point into that fusococcin binding groove. And in each case, you can see the wild type cysteine residue in yellow. So here, the cysteine residue that we're going to tether to is far away from the peptide. Here, it's quite close to the peptide. Here, it's actually overlapped. Peptides overlapped with that residue. So these 5 peptides vary in their overall shape of the interface, in the residues that are pointing towards the small molecule binding site. And we want to ask the question, how adaptive are these protein-protein interactions? I showed you that's really important for inhibiting protein-protein interactions. Is protein adaptivity also important for stabilizing protein-protein interactions? So we took these 4 different peptides as an example. We did 5, but here are 4. And we measured them all for binding of these fragments by mass spectrometry in the absence and in the presence of these client peptides. So here's an example of a molecule that's inhibited by the peptide. In the absence of the peptide, it binds about 70%. In the presence of the peptide, it's only 25% bound. So this quadrant, the compounds are inhibited by the peptide. In the yellow quadrant, the compounds bind neutrally with peptide. They bind the same whether the peptide is there or not. And in the red quadrant, this is the quadrant of interest. These bind more strongly in the presence of the peptide than in the absence. And when you look at these 4 different screens, you see some interesting things. One, there are some compounds like this green one that stabilize many different protein-protein interactions. And then there are others that are specific for each one of these different protein complexes. This one stabilizes target A. This one stabilizes target B, but has no effect on target A, and vice versa. So let's look at this pull back. How about any one compound? What do we see? So if you're not used to looking at these kind of heat maps, these are the 5 targets going across, and each box going down is a different compound. So we see that there are a small number of compounds shown here that are active against many different protein-protein interactions. Then there are sets of protein-protein interactions like CRAF, for example, that has a number of molecules that only bind to it. The lighter the color, the better the binding. So CRAF has a whole set of compounds that bind to it and to nothing else. Similarly, FOXO has a number of compounds that bind to the FOXO 14-3-3 complex, but not to any of the other 14-3-3 peptide complexes. So let's look at a couple of these. These are examples from the CRAF and the FOXO. In both cases, the peptide here, these are 2 CRAFs. The peptides are shown in this dark blue. And then this kind of indigo blue shows the molecules. And you can see a couple of things that I want to highlight. One is that these molecules really scope out the space in between 14-3-3 in white and the peptides in blue. And they're really sandwiches together. They're sandwiched together between the protein and the client protein, CRAF, and the 14-3-3. The second thing is that the peptide itself shows adaptivity. So the conformation of the peptide here takes a turn, whereas here it goes much straighter. Okay, so that it is indeed true that the protein-protein stabilization traps different conformational states of the client protein. And then finally, another example, this is FOXO is a peptide that binds to 14-3-3 with a steep turn. And here we find a completely different set of compounds that bind right next to. So like the first example with estrogen receptor, these compounds stabilize by acting more like gum, where they bind to 14-3-3 here, and they bind to the peptide next to it with a different face of the molecule. These are very potent stabilizers. These fragments coming right out of the screening hit can stabilize these protein peptide complexes by up to 250-fold. So really from a weak micromolar inhibitor to a very potent nanomolar, complex affinity to a nanomolar complex affinity. So we then optimize these compounds, and I don't have time to show you the chemical optimization right now, but I want to show you one of the biological results going back to estrogen receptor again as our example. So you'll recall that estrogen receptor binds to estradiol that induces dimerization, DNA binding, and transcription. Here we use a transcriptional readout that that codes for luciferase. And then if we have active estrogen receptor expression, then we'll express luciferase, and we can see that when we add the substrate luciferin as a glow, as bioluminescence. So this is just the assay setup, and these are some controls. So as along the x-axis here, we're titrating in estradiol. So as we add estradiol across, we increase the amount of transcription shown by luciferase. If we treat with fulvestrant, this is the ER degrader. It binds in the E2 ligand binding domain, so it competes with estradiol. Again, we see we have to add more estradiol to get any kind of signal because the fulvestrant competes out the estradiol. When we look at the natural product, fusicoccin, and its analog, NNAC, we call it, these also do a very good job of suppressing luciferase transcription. But this is not competitive with estradiol because these are binding in the 14-3-3 binding site, not in the estradiol binding site. And this compound is a negative control. It does not stabilize the 14-3-3 estrogen receptor interaction, and it doesn't inhibit transcription. So now we use 3 of our compounds. We have a negative control. So these are compounds that we identified with disulfide tethering and then optimized. This one was a bad optimization. It's not active. This compound has, see in this green line, has activity in the low micromolar range, and this compound is about 5-fold more potent. When we take these into the same luciferase assay, again titrating in estradiol, here's the signal of increasing transcription as we add estradiol. Fusacoccin is a modest suppressor. ICI is fulvestrant, that's a potent suppressor of estradiol-mediated transcription. Our negative control has no effect, but our two positive controls, in particular this most potent yellow compound, effectively suppress estradiol independent of the concentration of estradiol that's added. So in short, I've shown you how we can systematically identify protein-protein interaction stabilizers using 1433 and many of its client proteins as examples. These client proteins bind to the composite surface. They don't bind to 1433 very well. They don't bind to the target. They bind to the new interface created by the binding of the client to 1433. And by doing this, using a number of important drug targets, we'll be able to modulate these so-called undruggable targets, including a number of transcription factors and intrinsically disordered proteins. Just one point I want to make about 1433. You have to go looking for it, but when you look for it, it's everywhere. This is, probably can't read it in any detail, but this is a pathway, a common pathway diagram that you're used to seeing, probably where you have a growth factor receptor that binds to, say, epidermal growth factor. This creates an oligomerization, which then triggers phosphorylation inside the cell. Eventually, this phosphorylation cascade leads to transcription. In this MAP kinase, PI3 kinase pathway, 1433, here shown in light blue, binds to 18 different proteins in this cascade. So many of the phosphorylation sites that you see are clients for 1433. And one particularly interesting example is CRAF. We've been looking at the inhibitory site on CRAF. So residue 259 on CRAF kinase inhibits activity, inhibits downstream signaling, whereas binding of 1433 to a C-terminal site, site 621, activates the kinase by stabilizing the active dimer. So you can see it can be a very subtle thing. You really need to understand the 1433 client biology to know what it is you want to stabilize and what selectivity is important. With that, I'd like to thank Team 1433, my collaborators, Christian Altman and Luc Brunsfeld, both at the University of Technology in Eindhoven, my chemistry collaborators, Jeff Needs and Adam Renzo, both professors at UCSF, and our project teams. In red are the people who started the project. In blue, those who have taken up the mantle and who just did some of the work I described today, and then active members of the team whose work I didn't describe, shown in green. Last point to make is that the three of us started a company called Ambigon, and the goal of Ambigon is to really build on and commercialize these technologies for cancer and for other diseases that function by stabilizing 1433. And with that, I'm out of time, and so I'll be happy to stop and take any questions. Thank you very much.
Thanks, Michelle. Boy, farm chem is certainly not an area that I know a huge amount about. Please enter your questions in the Q&A section if you have them for this rare opportunity to ask Michelle. So I'm slightly lost. Can you remind me why that peptide is important? So you've got 1433. It's binding to its phosphorylated targets, and the peptide is there, and you're modifying the interaction by adding these small molecules. What was the where
does that work? Okay,
play?
yeah.
Yeah.
So the peptide is the model for the whole protein-protein complex.
Oh yes. Okay. Thanks. Yeah. You did mention that at the start.
Yeah, that's fine. I can go back and do that again. So that's because it's hard to work with the full-length proteins, hard to crystallize. So this way we can really, speed up the discovery.
Okay.
Let's see some questions. Can any 1433 support any target? or are there different forms of 1433? Okay, this is a good question. There are 7 isoforms of 1433, but they're very, very, very similar to each other. So whether they stabilize different targets, I'd say is a matter of some controversy. And for us, there are 2 basic approaches that we can take to stabilizing 1433 client complexes. We can either be specific for, there's one isoform that has that cysteine residue that we've been targeting. So those are isoform specific stabilizers. Molecules like fusacoccin don't have any covalent interaction with the protein. They're non-specific, so they stabilize all the isoforms of 1433. So the question is, do you want isoform specificity or not? And that's a question that we're seeking to answer. What areas might this be applicable to outside of oncology? Okay, so lots of good targets outside of oncology. FOXO, for example, is a metabolic target. It's a target for diabetes. And especially in neurodegenerative diseases, there are a number of interesting targets for 1433, and they fall into 2 basic classes. Many of the intrinsically disordered proteins that we associate with diseases like tau in Alzheimer's disease or tauopathy, and alpha-synuclein in Parkinson's disease, these are clients for 1433, and 1433 is involved in their degradation or their processing or their sequestration. So it could be that we want to stabilize those interactions or specific sites of those interactions. Also, some of the proteins that are known to be good targets, say in Parkinson's disease, LRRK2 is a kinase and signaling protein that's considered a good target for Parkinson's. It's activating mutations. So one of the reasons it's a good target, a validated target, is that there are mutations in the protein that lead to familial or early forms of Parkinson's disease. Those mutations function by inhibiting 1433 binding. So in the wild type, so that tells us that 1433 binding to LRRK2 inhibits LRRK2 function. Okay, so that makes that a good target. So a number of good ones. Okay, let's see. So the next question, did you say you mutated 2 additional positions to cysteine in addition to the native NER? And if so, why are 3 better? Okay, let me explain that better, Carl. So in 1433, so we're doing the disulfide tethering technique. That was the approach that we were taking. So we needed a cysteine residue. There's that wild type cysteine residue at cis38, and that was at the very end of the pocket. So we were just starting this work. We didn't want to be too smart. What if that's too far away from the peptide to stabilize that interaction? So we mutated that out to asparagine, and then we mutated in 1 at a time cysteine residues at different sites. So we're really asking the question of distance from the peptide. Are there different protein complexes that have different preferences for cysteine residues at different sites? So we just use 1 at a time. And the best one is the wild type, because that's the one we can turn into a covalent probe or a covalent drug, whereas the mutated ones, we can't make use of that in cells. How do you envision water molecules moving around or interacting with these new surfaces? Okay, hugely important question for people who think about medicinal chemistry. So proteins interact with each other and interact with molecules in the context of water. And water is often intimately involved in these complexes, for example, holding together 2 polar interactions. So water can act as a bridge, making hydrogen bonds with a molecule and a protein, and in a sense holding that protein together through the water molecule. There are also water molecules that you see in crystal structures that are in fairly hydrophobic regions of a protein, and they're not very comfortable there. So you can get energy by displacing what we call an unhappy water from a hydrophobic site. But we also see a number of interactions with our molecules that are water mediated. So sometimes we do displace waters, and sometimes we remodel the water interface to help us hold our compounds together. And then during optimization, we can replace that water molecule with an interaction that we build into our molecule itself. Can you comment on potential resistance mechanisms that might arise? Yeah, So 1433 is very rarely mutated in cancer. And you could argue that that's because there's been no selective pressure because there are no 1433 stabilizing molecules in the clinic. So I would say for our cysteine reactive compounds, removing the cysteine on 1433 could be a mechanism of resistance. You also see in cancer, you very rarely see the 1433 phosphorylation binding site on the client protein mutated. So BRAF, for example, you see activating mutations in the kinase active site. You don't see activating mutations in the phosphorylation sites that bind to 1433. Again, maybe because there's no selective pressure. So these are things we might see. Now, on the flip side, there are different mechanisms of resistance than we currently see with active site binding ligands that are in the clinic now. So the kinases, you get mutations in the kinase active site. Here we would get a different set of, our compounds would not be resistant to those mechanisms of resistance. And for estrogen receptor, it's the same. There are estrogen receptor mutations in the ligand binding site that make fulvestrant and other estrogen mimetics bind less potently. That doesn't have an effect on 1433 binding. Are there other types of proteins like 1433 that interact with proteins with promiscuity? Yes, it's interesting. So when I first talked to Alan Ashworth, who's the head of our cancer center, about 1433, he said, oh, I just thought it was like an SH2 domain. It just binds to everything. So SH2 domains are protein domains that bind to phosphorylated tyrosines. So the phosphorylated tyrosines bind to SH2, phosphorylated serines bind 1433. But then we managed to convince him that it was interesting and specific. So there are a number of domains that scaffold lots of different kinds of proteins that recognize phosphorylation sites or recognize SUMO or ubiquitin, other post-translational modifications. And we're also really interested in classes of proteins like WD40 domains. So these large classes of proteins that bind to a lot of different peptides and proteins for different functions. And Ursula, that's great. So many friends. Is the sulfhydryl group on the fragment or the disulfide, the thiol on the fragment just used for screening, and may it be removed in the later optimization, or do we keep it? If we have a wild type cysteine residues, I usually show this slide. So there are different ways that we can optimize our fragments depending on the surface and the needs of. Sorry. Okay, can you hear me again?
Yes.
Yeah, it just decided to use my headphones for no particular reason. Okay, so the sulfhydryl itself, or the thiol itself, is usually a dead end. We don't want to keep that around too long, but we can convert it into an electrophile, into a more potent cysteine binding group if we have a wild type cysteine residue in our protein. So sometimes we'll develop our molecules towards covalent drug leads or covalent probes, cellular probes. If it's an engineered cysteine residue, so if we've made that cysteine residue specifically to find a ligand, then we try to remove that, the necessity of binding to that cysteine residue. So we have a number of tricks that we're working on to use the tethering approach to find the fragment, but then we want to link fragments or optimize those fragments so they no longer need to be covalently bound to the protein. Thank you. There. Oh, one more.
I'm muted. Oh, good. There's another one.
Do you think there is a particular serial order to the binding? So different propensities or affinities to different orders, or is it all at once coming together? So this is a really important biophysical problem, because when we're stabilizing protein-protein interactions, we're essentially creating a three-body problem, which you may remember from physics is hard because all three things have to come together at the same time. So what we rely on is changes in residency time, so changes in the off rate. So the peptide or the protein-protein complex occurs and then it comes apart, comes apart. When it comes together, we trap it with the molecules. That's how I think about it. And then we slow the off rate. So then once the ternary complex is found, then it stays bound longer. And so that's why it has a higher affinity. The dynamics of all of these things coming on and off are difficult to measure, but I can say that we have different affinities of the interaction. So we have different intrinsic lifetimes for those interactions, but they are all long enough for us to find stabilizers that bring it down to a very slow off rate. So kind of it's a general question, but it is something that we're very interested in trying to get a better numerical approach to. So to experimentally determine the off rates and the order of interactions. So thank you. Okay.
Cool. No more questions?
That's great. Well, that was really an efficient way to answer questions. Thank you.
I'm glad people like questions because, man, this is really out of my sphere. I really enjoyed learning about it. Thank you so much, Michelle. I know everybody who attended appreciated it too. Thanks very much. See you around.
Thanks, everybody. Bye-bye.
Bye.